welcome to my poster, Barriers to Detecting Cell Type Specific Regulatory Signals in Gene Co-Expression. Gene co-expression analysis is something that is routinely carried out, often for the purpose of inferring intracellular gene regulation. But of course, bulk tissue samples contain multiple different cell types, and so any expression pattern that is particular to a cell type would likely undergo some dilution at the bulk tissue level. And so to explore and quantify the dilution impact, I'm using a combination of data simulations in this work as well as analysis of real transcriptomics data. And so here we propose a model that describes the relationship between bulk tissue expression profile and the underlying cell type specific expression profiles. Essentially, if you have a bulk tissue expression data set, any given gene can be represented as a expression vector, and we simply model that vector as the weighted sum of the individual cell type specific expression vectors that is underneath the bulk tissue sample. And so given this model and literature estimates of the cell type proportions to use as weights, as well as the expected levels of gene expression variability both within and across different cell types, we developed a simulation framework to generate single cell expression data, and then single cell transcriptomes are then put together to aggregated to form simulated bulk tissue expression profiles as well, and that way we can compare the two levels and assess the impact of dilution. And so here on the left hand panel, I'm plotting the amount of signal per gene that is coming from each individual cell type. And so, for example, the genes that are plotted on the right over here are mostly driven by signals from the astrocyte cell type specific expression vectors, but of course those are rare. You do have a lot of genes that are clustered at the left hand side here where most of the cell type specific signals are lost, and so this is true for most cell types. Excited neurons is a little bit different because it is the most abundant cell type, but still there's substantial reduction in the cell type specific expression patterns that make it through to the bulk tissue level. And so this directly translates into dilution of co-expression signals. And so on the right here, I'm showing this cell type specific expression pattern that is particular to exaggerated neurons that is then completely diluted out at the bulk tissue level in the presence of all the other cell types. You do have exceptional cases where if both genes are highly variable relative to the other cell types in the cell type that they are co-expressed, and you can have that preservation of co-expression signal at the bulk tissue level. But like I previously alluded to, those are likely pretty rare. And so more directly, here's the overall look at the co-expression, cell type specific co-expression signals. So those are specific to microglia because microglia only accounts for about 10%. For 10% exactly, actually, small proportion of cells in our model, a lot of those co-expressions are completely lost in the bulk at the bulk tissue level. In excited neurons, again, it's a little bit better, but still a substantial reduction in co-expression signal. But what's kind of interesting is that even the co-expression signals that are shared among multiple different cell types, those would still undergo some dilution. They're not guaranteed to show up at the bulk tissue level, and so that's what's been plotted here. The reason for that is you can have situations where the two genes, one of them may be primarily driven by one cell type, in this case microglia, and then another gene in this case primarily driven by astrocyte. Unless they are intercorrelated between the two cell types, that co-expression signal would be lost even if it is present in both dominating cell types. And so at least in principle, this is something that can happen. And so even if a co-expression signal is shared among multiple different cell types in the bulk tissue sample, they're not guaranteed to show up at the bulk tissue level. And so to explore further, we expanded this analysis to looking at a real data set. And so this is a data set that was produced by Matthews et al. In 2019. It has both single cell and bulk tissue data generated from the same subjects, which makes an interesting case that we can do a similar analysis on as we did for the simulation. And so we took this data set and we discovered 167 gene co-expression clusters in the exciter neurons alone, and then we looked at the cluster integrity of those clusters at the bulk tissue level, and you can see that there is this pretty substantial reduction of cluster integrity. However, you do have this preservation of clusters of genes that are particularly enriched for ribosomal protein genes. So these are the highlighted clusters over here. And so the ribosomal protein genes appear to have preserved their co-expression somehow. We did a little bit of analysis to confirm that those signals are indeed coming from the underlying cell type specific expression patterns. In the interest of time, I won't go into a lot of details, but essentially if you remove the cell type specific expression patterns, then you remove co-expression among the ribosomal protein genes as well. And so there's something exceptional and particular about this group of genes that makes it resistant to dilution. One possibility is that they're all coming from the same cell type, which is not true as in this heat map. And so another hypothesis that we have is that they actually have a lot more intercell type correlation compared to other genes, which is actually turned out to be the case. And so in summary, we're able to show that dilution likely removes a lot of the signals. Ribosomal protein genes are exceptional to this rule, but their co-expression may be preserved by this intercell type synchrony across the samples. And we're actually able to corroborate some of these findings in another data set.